Hello and welcome to the Clinical Care Options podcast series, a closer look at evolving treatment strategies for rheumatoid arthritis. Today's episode, taken from a recent symposium, is on targeted DMARDs. Features Dr. Stanley Cohen, Dr. Sheetal Desai, and Dr. Eric Ruderman. If you want to follow along with the slides, just go to the show notes and click on the link. You can also find a link to additional online education on RA from Clinical Care Options. Now let's get started and hear about targeted DMARDs, starting with Dr. Eric Ruderman.
So we're going to look a little bit backwards here. The title of this whole symposium is, Evolving Treatment Strategies for Rheumatoid Arthritis. And before we spend some time talking about where we are now and where we're going, I think it's interesting to look at where we've come from. So imagine you have a patient, a 30-year-old woman who has swelling and synovitis in her PIPs or MCPs in her wrists for about 3 months, and you've concluded she has rheumatoid arthritis. What are your options? And going backwards, not so very long ago, it was mineral baths. Not too long after that, it was looking for an infectious etiology for rheumatoid arthritis, which is actually where sulfasalazine came from. And then in the 40s, we had the brilliant leap ahead to inject a toxic heavy metal into people, which was really the standard of care for many, many years, into the 60s when hydroxychloroquine came along, and really until the early 80s when methotrexate arrived. The 1990s was an era of conventional synthetic DMARDs, methotrexate, sulfasalazine, leflunomide not too long after that, and hydroxychloroquine, and the infamous triple therapy in there. And by 2019, we had gotten to a place pretty clearly where methotrexate is our first line, and now 2020, our first line therapy for nearly all patients. And then within a few months, in the era of treat to target, adding additional therapy, targeted therapy, either a biologic or now a targeted small molecule if patients had not achieved their target. And in terms of those targeted therapies, here's where we've come in the last 20 years or so. The very first molecules to hit our closets to be able to treat patients were etanercept and infliximab, TNF inhibitors, later additional TNF inhibitors, and then in the early 2000s, we started to get other non-TNF biologic therapies, targeting B cells, T cells, interleukin-6, et cetera. And then in 2012 came the era of the targeted small molecules, or the JAK inhibitors, with the first being tofacitinib, and subsequently baricitinib, upadacitinib, and now filgotinib, which has been approved in the EU, but is still under review in the US and continuing trials. The key difference with these targeted small molecules is where in the body they work. And so we're used to using biologic therapies that work extracellularly. These are, as we all know, large, complex molecules that can't be given by mouth. They have to be given parenterally because they aren't absorbed. And they work to block cell signaling between cells. The kinase inhibitors take advantage of the fact that when cytokines engage a target cell, they initiate a signaling cascade that transmits that signal down to the nucleus of the cell, the business end of the cell, where proteins are produced. There have been a number of these signaling cascades described, and there have been attempts to harness some of these to treat rheumatoid arthritis over a number of years. The first was probably the MAP kinase pathway that just didn't pan out. Syk kinase pathway, which also signals for a bunch of different cytokines, also didn't pan out. And we finally hit the place where the target that seems to have been most successful has been the Jun kinase pathway, or JAK pathway. Looking at this in a little bit more detail, the JAK signaling pathway is shown here in a fairly simplistic cartoon. The concept is that there are four different JAK molecules within the JAK family: JAK1, JAK2, JAK3, and no, not JAK4, but TYK2 for a variety of reasons that aren't worth going into now. The way these work is that when a cytokine engages the receptor on the cell surface, two separate JAK molecules form a dimer on the intracellular portion of that cytokine receptor. And typically, those are heterodimers. They're not always the same JAK, and I'll show you that on the next slide. That dimerized JAK complex becomes phosphorylated, and in turn phosphorylates a series of STAT molecules, signal transducer and activator of transcriptions. These are the molecules that transmit that signal from the cell surface down to the nucleus. Those STAT molecules dimerize as well. They get phosphorylated, they go down to the nucleus and enter the nucleus, at which point they initiate gene transcription. Here's another slide that shows a bit of the specificity here, although not in full detail. And the idea is that different cytokines will signal through different members of the JAK family. And what you see here is a couple things, that there are different JAK dimers, different JAK heterodimers that are involved in signaling for the various cytokines. And importantly, it's typically a heterodimer. The one exception being JAK2 homodimers, which are involved in important signaling pathways for hematologic parameters. And that actually may have some relevance to some of the toxicities that we've seen with JAK inhibitors over the years. There's been a lot of talk about selectivity of JAK inhibitors. And I need to clarify that by saying that any selectivity that we know about is really selectivity in vitro. It's selectivity in assays in the lab. We really don't have a solid insight into the selectivity in patients who get these JAK inhibitors. But in vitro, at least, there's some suggestion that tofacitinib is relatively selective for JAK1 and JAK3. Baricitinib is more selective for JAK1 and JAK2. And upadacitinib and filgotinib are most selective for just JAK1. What's listed on this slide are the 5 JAK inhibitors that are currently available in the world. The first 3 have been approved in the US, and you can see their indications there on the right. The 2 of them for methotrexate inadequate responder patients and the baricitinib for TNF inadequate responders. Filgotinib was just approved in the EU in September, but it is not approved in the US, pending further studies. And then pefacitinib, which is a unique JAK inhibitor that targets a number of different JAK molecules, has only been studied and approved in Japan and Korea, and is not likely to show up in the US. So I'll turn this over to Dr. Desai at this point, because I think she's going to lead us in a little discussion of what all of this actually may mean.
Thank you, Eric. So that was a really great review of the JAK inhibitors and the different subtypes of the JAK inhibitors. Eric, do we have any preclinical data with JAK inhibitors that can help us inform clinical decision-making?
I don't think so. I mean, I think that's been a challenge. There's preclinical data that, at least in the lab, shows differences in selectivity. And there's been people have postulated over the years that may affect efficacy, that it may affect safety. But there hasn't been a tremendous amount of data in animal models that would suggest that we can pick and choose based on that. And what's been really, both interesting and frustrating, depending on which side of this you look at, the selectivity, at least the in vitro or preclinical selectivity, hasn't really translated into any significant differences as far as we can tell in either efficacy or safety in patients.
So the JAK selectivity that you discussed really doesn't impact efficacy or safety either?
I don't think so. I mean, what we learned from preclinical data, are some issues in terms of absorption and some issues in terms of metabolism, but it doesn't seem to help us once we get into patients understanding differences in efficacy or safety. Stan, I mean, you've been using these drugs for a while, and tell me if you've found differently, but I just haven't seen that.
You're right. So the original intent is that you didn't want to knock out JAK2, because basically the genetic models, the animals, that was lethal with no erythropoietin activity. So there was the desire to try to impact JAK1. JAK3 is a major component of the kinases in immune cells, lymphocytes, and so forth. So it was an attractive early target. But the FDA considers tofacitinib and baricitinib pan-JAK inhibitors based on the data. And the preclinical assays are difficult. There's enzymatic, biochemical, there's cellular assays. It depends on how much substrate you add to the assays, how much ATP, and it depends if you're using the Cmax or the average concentration. You can get different results. But again, as development goes on, they are trying to be more selective, have less off-target impact on other JAKs and other kinases. But to date, as we show you the clinical data next, the preclinical selectivity has not really made an impact on either efficacy or safety.
Thank you, Dr. Cohen. To hear the next episode in this series, click on the link in the show notes.